my talk for deep chemistry, protein folding, and molecular modeling in Wolfram language. That means we are going to dive today into a bit of biology, chemistry, and of course, artificial intelligence. The outline for today is, well, we are going to see what is the importance of the structures of the proteins and how we go from sequence to structure. We will see a bit of history of the protein structure discovery and how this helped the scientists to train machine learning models like AlphaFold, and what was the big revolution about this new model, and how did we get from AlphaFold to ESM-fold. ESM-fold is a meta model that is faster and can run in almost every local machine from users, and how we bring ESM-fold into the Wolfram ecosystem via ONNX format files. And we will dive a bit in the use cases, like how to discover patterns hidden in protein sequences, how to use the ONNX files models from ESM-fold to create a pipeline for discover protein structure, and how to use biomolecule function in the Wolfram ecosystem to model protein structure. So from sequence to structure, this is a journey of protein folding and AI. The proteins are everywhere in life, and they are the machines of life. That means everything, all the functions in your body are determined by how these proteins function and what is its shape. So every protein has a function, like for example, in your blood cells, the red cells, how many atoms of oxygen it can carry depends of the shape. If this shape of your blood cell is not correct, it will make that your blood cannot carry correctly the oxygen. And there are many diseases that are caused for a failure in the way this protein, the shape of these proteins, for example, Alzheimer, and something like the cow, crazy cow disease is also caused by a failure in how this protein fold in its shape itself. So predicting this shape is very important in biology, since it has been a problem that comes from many years ago. And this structure of the protein is determined by the atoms that conform it, like the sequence that shapes the protein. So this is determined by the amino acids that you can find inside, and how they interact with each other. Inside you can see, for example, hydrogen bonds and distinct physics interactions that makes them attract or repel to each other. So how do we start studying these shapes and how we discover how they fold? We can start like in the 50s from the last century with the Dr. Kendrew, that he modeled the molecule myoglobin using crystallography methods. So he took the myoglobin, made crystals from it, and then with crystallography methods, he got the first structure from proteins, experimental. And from this discovery, scientists started using crystallography to get the shape of the proteins experimentally. But obtaining this structure from crystallography is very expensive, around $10,000 per protein, and it takes a lot of time. Usually, one PhD, complete PhD, to discover just one shape. So, around the year 68 of the last century, we have this scientist called Affinsen, and he made his principle, like his theory, about that the sequence alone is able to determine the protein's shape. And from this principle, scientists from DeepMind developed AlphaFold, the first model that was able to machine learning model that was able to find the structure of the proteins, and then ESM-Fold from Meta. So, what was the revolution? Well, AlphaFold was the first model to win the CAPS challenge for predicting protein structures. And it was very impressive, impressed all the scientists around the world, because it was the first machine to get these results. But they weren't happy just with this. So in year 2020, they improved their model, and they got accuracy similar to experimental accuracy. And that was all a revolution into the biology area. Yeah, so after they made this, we were able to crack the folding code. And what decades of physics couldn't solve, we could achieve in months. Like, in all these years, using crystallography and other methods, we found around 150,000 distinct protein shapes. But after the discovery of this machine learning method with AlphaFold, we were able to discover 200 million new protein structures. So it was a huge step from what we had before and what we have now. And this opened the doors for a lot of improvements in science, like in biology, in medicine, discover new treatments for different diseases, and so on. So what was the problem with this AlphaFold is that it was too huge, too big for running in local machines. Like, it needs not only the input sequence, but it also needs a database using multiple sequence alignment that fit each other together with the structure, the structure itself from the sequence, and the database from the sequence that we know. And the model processes the database and the sequence itself, and they fit each other in the Evo former. And then the structure model takes this processed data to reconstruct the protein structure. So it needs to process for each protein that we want to infer the structure. It needs to process thousands of evolutionary sequences, and it needs to process the sequence itself. So it takes a lot of computing resources and some hours to process. And also you need to have huge machines to run the model. So what Meta did, inspired in AlphaFold, was create this new model called ESM-Fold, which has a language model that was trained only with sequences, and the sequence were masked in the input, and they pre-train the model to predict what elements or atoms in this sequence that was masked, what was not there, to predict what was masked. And this pre-training method allowed the model to know how the sequence are constructed. What is the correct sequence order in every case? And after that, they created a similar method like in AlphaFold to process these embeddings, and then with the same structure model that AlphaFold used, reconstruct with these embeddings the 3D structure. And removing the multiple sequence alignment, they skip 90% of the process that AlphaFold needs to do, reducing the computing resources needed and also time. This ESM-fold runs in seconds, while AlphaFold needs hours to run. And without losing too much accuracy, like you can see here, ESM-fold achieves 83% of accuracy, while AlphaFold achieves 88% of accuracy. So without losing too much, you are gaining a lot. So you are saving time and computing resources. But how do we use these models in Wolfram language? There is some framework called ONNX that allows interoperability between frameworks. So when you have a model, for example, in PyTorch, TensorFlow, or Keras, you can export this model to ONNX format, and then these frameworks are not only capable to export into ONNX, but also import from ONNX. And this is the case for Wolfram. So what we did was exporting the model from Meta to ONNX, and our framework is able, using just import function with the next external object option, to import the model. So here you have the parts of the model necessary to run the pipeline. You have the language model, you have the folding trunk, and you have a post-processing part that takes these outputs from the folding trunk and makes some process to make it able to reproduce the 3D structure. So after exporting these models, the language model and the folding trunk, you can play with it. For example, you have here an example of how to use the language model, which, as I said, create embeddings from each sequence. You can use this model to make clusters from proteins that have something in common. For example, here I look for enzymes and structural proteins and listed them, then join them together to not tell the model what sequence belongs to what group, and then just use the ESM language model to encode this sequence into embeddings, which is a multi-dimensional space, and then just use it in the function feature space plot with this feature structure to plot it and see what was the result from these embeddings that we obtained from the model. And as you can see, the red dots fall apart from the green dots. That means the model was able to identify what was the enzymes and the structure molecules, proteins. But how do we join together these models from NNX? As I said, we have 3 parts that I was able to export from the project, from Meta project. It was the language model, the folding trunk, and the post-processing part. So first, you have a sequence. You encode it. That means you transform this sequence into numbers using a dictionary. So every letter in the sequence corresponds to a number. And this is what we need to feed into the language model. And this language model will convert it into multidimensional embedding space, which is just a matrix of numbers. And we will use this result to feed the folded trunk here. Sorry. This is just initializing, for example, the mask. The mask doesn't exist, so we don't want to mask in this case because this is only for training. So we create a matrix with ones, which means do not mask. And we initialize the state of the recycling, recycling part. This recycling works like for refining the result. But in this case, we are not going to do the iteration. We are going to run it just one time. We don't want to refine. We just want to see the overall pipeline. So after we get this structure using the folding trunk, we pass the result into the post-processing. And what we will get, we need to convert it into the PDB file. This PDB file has been used in the different database to describe a molecule, these protein molecules. So we need to convert what the model gives into this PDB file. So we can interpret this result into a structure from a molecule. Once we get this PDB file, as you see here, we use the function import string. We identify the text as PDB and that we want a biomolecule to be displayed. And here you have the biomolecule. And then use biomolecule plot 3D to graph this biomolecule. And here you can see in Wolfram, we can just dynamically see what is the result. And this is basically the pipeline. Maybe I missed to say that, as you can see here, we are not only using the ONNX files, but we need to study the project to add the code that is missing or the inputs that the model needs in order to run. That's something that we need to do always because, or almost always, because ONNX is not exporting the whole project from the code. Is exporting just the model or the parts of the model that we need. So if this is the case, in the case you want to use the model for custom applications, but in the case you want to only plot the structure of a molecule, you don't need to know the pipeline. You don't need anything of this because we have packaged all of this into the function called biomolecule. Here you have the reference for the biomolecule plot, for example, and you can look for the reference here for biomolecule function. And also we have published the ONNX files into our neural net repository. So in case you want to use the ONNX files, you can get them just by calling netmodel and the name of the model. And here you can use it for custom applications. But if you don't want, just use the biomolecule and the biomolecule function, and everything will be packaged for you. And you have two options. For example, if you use biomolecule only alone without this option, it will call an API that will give you the protein structure from the API call. It will infer it from the model. But if you want to use it locally and you don't want to depend on your internet connection, for example, or you want to keep the model in your computer, you call it like using this option, biomolecule folding method local, and then you will have the ONNX files in your laptop. So using this biomolecule, I'm using two things. I'm calling the biomolecule from a PDB file, and this PDB file has the experimental structure. This is what the molecule should be, and this is what the sequence from this molecule is. So I'm passing the sequence only, and biomolecule is predicting from my local model what the structure should be, and this other is the theoretical or the experimental shape of the molecule. So here, if we compare experimental versus predicted, you can see how they match together. So this is the end of my presentation. Thank you for being here, for attend, and I hope you have enjoyed. Thank you very much. Is there any questions? Get the parameters. I think yes, because here you have both things are biomolecules, and you can compare them extracting the residues and the atoms. I don't know if Jason can answer this because he's the expert into the biomolecule package.
Let's see, what do we, what was the question here? Let's see, so I mean, can you hear me?
Yes, I can hear you.
Okay,
Yes,
so I don't know about so can the amino acids be altered? Not that I know of. So most of the models out there really assume that you have the standard amino acids. Yeah. And then I saw a question asked about, what was it in the chat? About printing, 3D printing, right? And so we have, so you can take one of these biomolecules, whether it's a folded one or one experimental, and pass it to the function called molecule mesh and make a 3D mesh for that. And then you can, yeah, you can print that. I've never actually, I don't actually have a 3D printer, but I imagine you can. And right, I mean, protein complexes, like multiple ones living in the same space. ESMFold does have some capability to do that. Essentially, I think what they do is just write in a big gap in the middle and then let the thing fold and then take the gap out, and so you're left with two pieces. I don't know how physically relevant that is to the actual protein complex, to be honest. Yeah, and so I don't really know how well these models do for simulating complexes or predicting complex structure, multi-chain structure. Right, and so yeah, and that's to the next question, right? multiple polypeptide chains. I do not know that even that we are there yet, but we're always trying to keep up with the literature.
Someone here asks, does AI allow for amino acids to be structurally altered to determine folding structure? I think you are allowed to pass any sequence to this model and see what happens.
Right.
About the quaternary structure, I don't know, and I'm not a biologist.
All right. Yeah. So I was kind of answering out loud, but I can put those as a respond here. Multi-chain complexes are not very well modeled at the moment by ESM fold or AlphaFold.
Is there anything similar for nucleic acids?
Not that I know of. If you find something, let us know.
Now that's interesting. What is the limit of sequence length that can be folded? I'm not sure, but it has a limit.
Well, the server, like when you do it using the API, there's a limit of 400 residues, but I believe, or it might be 200. I do believe it's 400. But when you're running it locally, I think it's just related to the amount of time you're willing to spend and the computer you're running it on. If you have a GPU, you'd be a lot better off if you're trying to do something large.
Mm-hmm. Here, the model that I import from ONNX, it doesn't have like the file doesn't show any limit, but I'm sure it has it. It should have it. Like when you try to run, I don't know, 2000 amino acids, it will give through an error, I'm sure.
Right. It might try and create some internal array with a greater than 32-bit integer number of rows or something at some point. But in general, I think it's just related to how much time have you got to spend.
Have you ever had one of your proteins created at a place like any synthesis? This is like generative model, because I saw also, I think it was DeepMind, the same from AlphaFold or something similar, that they won the Nobel Prize, but it was for a generative model.
Let's see, why is ESM fold so much faster? Correct me if I'm wrong, Siri. I think it's the fact that you don't have to do any kind of multiple sequence alignment, no lookup in a large database. And
yeah, basically AlphaFold, they were using two inputs: the sequence itself that you want to study, and also the whole, or a huge matrix with the evolutionary state, like information from other sequences that may be similar to the one that we are trying to predict. So by using that, they are inputting this huge matrix together with the sequence that you want to predict the structure. So for that, you need a lot of resources and process a lot of information. So it makes the model too slow. And yeah, ESM4 drop this process from from this huge matrix that gives extra information from the outside, and just use a language model to pre-train over the sequences itself. And this language model is used as a feature structure. Protein interactions. No.
No, but you should visit Bob's talk later in the day, Robert Nakbar's talk later on, where he's trying to get into doing actual protein modeling in the Wolfram language using the biomolecule.
Mm-hmm. Yeah, this model is used only for predictive structure. That, well, this is a lot for science. This was a lot for scientists because before they needed to do crystallography to discover exactly what was the shape of the protein, and now we have reached almost experimental accuracy, and you don't even need to enter to a lab. That is a huge step.
Thank you for speaking, Siri. Siri, this was a really good talk.
Thank you.